Great. I'd like to spend the next few minutes giving you a brief background on chimerism testing. Then I'll introduce you to our technology for isolating different cell populations from blood. I'm then going to focus on case studies from 3 different HLA laboratories across the United States who've each brought chimerism testing in-house. We're going to highlight the Florida Hospital Tissue Typing Laboratory in Orlando, Florida, the Children's Hospital of Philadelphia, and the Stanford University Blood Center. Now, each lab uses RoboSep in a different way to isolate cells for downstream chimerism analysis. And finally, I'm going to end up by opening up the discussion to our panelists. We have Kurt Lind from the Children's Hospital of Philadelphia and Wendy Leong from the Stanford University Blood Center. So what is a chimera? Well, in ancient Greek mythology, it was a fire-breathing monster with the head of a lion, the body of a goat, and the tail of a serpent. In the context of hematopoietic cell transplantation, it refers to the presence of donor cells in the recipient after transplantation. Now, complete chimerism is defined as having greater than 95% of the hematopoietic cells being of donor origin, while mixed chimerism is defined as between 5 to 95% of the cells being of donor origin, or reciprocally greater than 5% are of recipient origin. So donor and recipient cells in a patient can be distinguished by testing informative genetic markers using one of a number of very well-established methods. For example, there's restriction fragment length polymorphisms, in situ hybridization using XY probes, and more recently, STR analysis by PCR sequencing or real-time PCR. Now, these methods have been discussed in many excellent reviews, such as this one by Bader and colleagues, and as such, I won't discuss them further here today. So why do chimerism testing? Although somewhat controversial, there's a growing body of evidence that suggests an association between engraftment kinetics over time and transplant outcome. For example, there was a nice paper published a few years ago by a group in the Netherlands showing that patients who were complete chimeras had less relapse and better survival compared to patients who were mixed chimeras or had an increase in the recipient-derived cells. So what physicians really want to know is what is the percentage of recipient cells present over the transplant period. They can then use this information as a predictor of transplant outcome. That is, to identify patients who might be at risk for relapse, rejection, or acute graft versus host disease. This then allows for early treatment intervention. For example, mixed chimerism in the T cell compartment could infer the patient will relapse. Now, knowing this information gives the transplant doctor time to either decrease immunosuppression so that T cells in the graft might recover and start to fight the cancer cells, or to give additional donor lymphocyte infusions to increase the graft versus tumor effect. Now, of course, it's much more complicated than this in real life, whereby the clinical context is critically important in the interpretation of chimerism test results. So is it necessary to isolate different cell populations for chimerism analysis? Currently, chimerism testing can be performed on DNA isolated from either whole blood or whole bone marrow, or from lineage-specific cells such as T cells, myeloid cells, and NK cells. Now, which source is required largely depends on the type of pre-transplant repertoire the patient receives. For patients receiving myeloablative transplants, isolation of DNA from whole blood may be sufficient. For patients receiving non-myeloablative conditioning regimens, some degree of lineage-specific cell isolation is likely warranted. And this is because patients can show complete chimerism in one cell subset, such as the T cell compartment, but can be partially or wholly mixed chimeric in another, such as the myeloid compartment. and these differences can have clinical relevance. In addition, using cell subsets can increase the sensitivity of the assay. So where a typical STR analysis might have a sensitivity of 1 to 5% on whole blood, the sensitivity will increase if subsets such as T cells or myeloid cells are used. So while not all labs isolate cell subsets for chimerism analysis, the use of lineage-specific cells is growing. If you're going to isolate cell subsets for chimerism analysis, such as T cells, myeloid cells, NK cells, you want a cell isolation system that gives very high purity and does not allow for cross-contamination between samples. Our RoboSep system does just that. With our technology, target cells are specifically labeled with dextran-coated magnetic particles using a bispecific tetrameric antibody complex, or a TAC. These complexes recognize both the dextran-coated particle and the target surface antigen. Magnetically labeled cells are then separated from unlabeled cells by placing the sample in a specially designed magnet. RoboSep is gentle on cells. The cells have excellent viability. It's quick. Most isolations take less than an hour to perform. And best of all, the reagents can be used at room temperature or 4 degrees and are stable if they're left out on the bench for an extended period of time. So the real advantage for a chimerism lab is that you'll rarely have to throw out expired reagents, and you'll have to validate your reagents less often. One of the best things about RoboSep is that it can be automated with a machine called RoboSep. RoboSep is a pipetting robot that automates both the cell labeling and the cell separation steps. It uses RoboSep reagents and procedure and doesn't use columns. RoboSep can isolate up to 4 independent samples simultaneously. And by independent samples, I mean in quadrant 1, you could isolate T cells from whole blood. In quadrant 2, you could isolate B cells from PBMC, and so on. The revolving carousel has places to put your magnet, the sample tube, reagent tubes, etc. And the cells are labeled and transferred to the magnet by the rotating pipetting arm. Probably the most important feature of the RoboSep for a chimerism lab is that each sample on the machine is handled by a unique set of disposable filter tips. This completely prevents cross-contamination between precious patient samples. The RoboSep itself is very easy to use. You simply choose the protocol you want to run from the protocol list, then load the carousel according to the on-screen prompts. When the carousel is loaded, you simply press the green Run button and you walk away. Depending on the protocol you are running, it will take between 25 to 60 minutes to isolate your cells. And when the run is complete, your isolated cells are ready for DNA extraction. The main advantages of RoboSep for a chimerism lab are that it saves technician time, as 4 samples can be processed in about an hour with only 5 minutes of hands-on time. It avoids sample contamination, as each sample is handled with a unique set of disposable tips. And all the steps are fully automated. There's minimal handling of potentially hazardous samples. So I'd like to spend the rest of the presentation highlighting 3 immunogenetics laboratories who've brought chimerism testing in-house. All 3 labs use RoboSep to isolate cells for chimerism analysis, but interestingly, they've all taken very different approaches to how they use the instrument to do this. In addition, the labs we're showcasing represent a good cross-section of the labs out there, from the Florida Hospital Tissue Typing Lab and the Children's Hospital of Philadelphia, who process a relatively small number of samples per month, to the Stanford University Blood Center, who process literally hundreds of samples per month. I'd like to thank all 3 laboratories for sharing their experiences with us, and of course for allowing us to share their experiences with you. We're very fortunate, of course, to have Kurt Lind from the Children's Hospital of Philadelphia and Wendy Leong from Stanford University Blood Center standing by to answer any questions you might have after the presentation. The first lab I want to highlight is the Florida Hospital Tissue Typing Laboratory. All the information I'm going to present today was kindly provided by Stephanie Corliss, the assistant manager of the lab. The Florida Hospital Transplant Group, and hence the Tissue Typing Lab, services the local Orlando area. Up until now, they'd been sending any samples that required cell separation out to a reference lab to be processed. But when they ran the numbers, they projected that they were going to spend greater than $200,000 sending samples out for chimerism testing. As a result, they decided to bring chimerism testing in-house so that they could capture some of this revenue. Since then, they've validated the cell separation aspect of the chimerism assay using our RoboSep instrument. The Florida Hospital Lab isolates 4 different cell types. They isolate T cells, B cells, myeloid cells, as defined by being 33 positive and 66 B positive, and NK cells. They process between 2 to 6 samples per week, and they either receive 8 to 10 mils of blood or 1 mil of bone marrow aspirate to work with. They require greater than 85% purity for all their isolated cell populations, and they don't require a large quantity of DNA for their downstream chimerism assay, which in this case is the ABI identifier STR kit. Prior to cell isolation, the Florida group concentrates their 8 to 10 mil blood sample using a hetastarch agent called RoboSep. Now, when RoboSep is added to the blood at 1 part RoboSep to 5 parts blood, it causes the red blood cells to low or form long chains, which basically just makes them heavy. These heavy red blood cell chains sink to the bottom of the tube, displacing the nucleated white cells into the plasma layer. When the mixture reaches a 50-50 interface, so you've got 50% red cells, 50% nucleated cells, the nucleated cells are harvested, washed, and resuspended in 1/10 the original volume. At this point, the cells are now ready for cell isolation using EasySep or RoboSep. Now, as this group only starts with 8 to 10 mils of blood, and they need to isolate 4 different cell types, we recommended that they try a sequential separation approach. So, from their RoboSep treated sample, B cells are isolated in the first quadrant of RoboSep using the CD19 positive selection kit. The CD19 depleted fraction is then transferred to the second quadrant, where T cells are isolated using the CD3 positive selection kit. The T cell depleted fraction is then transferred to the third quadrant, where myeloid cells are isolated using the CD33 66B kit, and the depleted fraction is again transferred to quadrant 4, where NK cells are isolated by negative selection. So this group is able to isolate B cells, T cells, myeloid cells, and NK cells in about 2.5 hours from a single sample. When we asked Stephanie why she liked the RoboSep, she said first and foremost was because it gives excellent purity. This is some purity data that she provided for 18 patients on the 4 different cell types that they isolate. As you can see, purities are about 98% for T cells, close to 100% for your B and myeloid, and about 97% for the NK cells. In addition, Stephanie mentioned how they like how easy the machine is to use, the fact that it requires minimal hands-on time, and allows them to isolate their 4 different cell types from a single sample. Chimerism testing was relatively easy for them to set up. They validated their assay on 70 samples, and the validation only took a few weeks. In terms of whether chimerism testing is revenue generating for them, they're still in the process of setting up the assay, but they do expect this to be the case. The second lab we'd like to showcase is the Immunogenetics Laboratory from the Children's Hospital of Philadelphia. I'd like to thank Kurt Lind for kindly providing us with the information I'm going to present. And again, Kurt has generously donated his time to be with us today to answer any questions you may have regarding his group's program. The Children's Hospital of Philadelphia is a large pediatric hospital that services a fairly big catchment area. The immunogenetics lab started engraftment testing in-house about 2 to 3 years ago, and they started lineage-specific chimerism testing about 1.5 years ago. They validated the cell separation portion of their assay using the manual EasySep system, and just recently purchased a RoboSep to automate the procedure. They mostly isolate T cells, but have started isolating NK cells in recent months. They occasionally isolate myeloid cells and B cells. About 25% of the chimerism tests they perform require the use of isolated cells, but Kurt indicates this number is growing. They perform about 4 CD3 T cell isolations per week and 2 CD56 positive NK cell isolations per week. They start with 4 to 8 mils of blood, that's EDTA treated, as they find this anticoagulant works best with their downstream DNA analysis. Now, if the samples have a normal white blood cell count, they'll isolate the cell subsets directly from whole blood. However, if the lymphocyte count is low, they'll phycol the blood and isolate cells from the PBMC fraction. Like the Florida group, they require greater than 85% purity for each cell population. They require very little DNA from each isolated cell subset, as the STR assay they run is very robust. But Kurt mentioned that they do sometimes need to run the DNA undiluted if it's an early post-transplant sample that doesn't have very many lymphocytes. And for downstream chimerism testing, they're using the ABI Profiler Plus kit. At the moment, they split their sample and isolate either T cells or NK cells, either directly from whole blood or from the PBMCs if the white blood cell count is low. Kurt likes the EasySep RoboSep system because it gives good purity. Below is some purity data kindly provided by Kurt from 10 individuals, and as you can see, purities are all above 96%. In addition, the cells are flow cytometry compatible, which is critical for this group as they assess purity on every single sample. Kurt kindly provided results from a typical chimerism experiment. So from 4 mils of EDTA blood, they phycalled it to get the PBMCs. T cells were enriched using the CD3 positive selection kit, and the purity was assessed at 99% by flow cytometry. They isolated DNA from the sample and obtained just over 8 micrograms of DNA. Chimerism testing only required 2.5 nanograms of DNA, and the percent chimerism was 71% for the CD3 fraction, but only 32% for the non-enriched fraction. And again, this highlights the differences in sensitivity using isolated cells versus whole blood. This slide just shows purity assessment for this particular patient sample. As you can see, the sample is quite clean and the purity is very good. Kurt said that the chimerism test was very easy to bring in-house. They validated the assay itself on 40 to 50 patient samples in parallel with another institute, plus they participated in 2 proficiency tests. They validated the cell separation portion of their assay on 10 patients by flow cytometry. Kurt indicated that chimerism testing is indeed revenue generating for his lab. The last lab we want to showcase is the Histocompatibility, Immunogenetics, and Disease Profiling Laboratory at the Stanford University Blood Center. I'd like to thank Wendy Leong for kindly providing the information I'm going to present. We're very fortunate, of course, to have Wendy as one of our featured panelists, and she and Kurt will be happy to answer any questions you might have after the presentation. The Stanford University Blood Center primarily services the local Bay Area, but they do occasionally process samples from other parts of the state or out of state. The HLA lab brought chimerism testing in-house a number of years ago to support the very large adult and pediatric transplant program, which includes both myeloablative and many non-myeloablative transplants. They initially validated the cell separation portion of the assay using a competitor system, but switched to RoboSep a number of years ago. At the moment, they have 3 RoboSep instruments that they pretty much run all day, every day, to keep up with the demand for isolated cells. This group isolates CD3 cells, 19 cells, 15 cells, and 56 positive cells from every patient sample, and they occasionally isolate CD34 progenitors. They process between 2 to 16 patients per day for all 5 subsets, which works out to between 10 to 80 cell separations per day. Wendy looked back at the previous year and noted that on average they perform about 500 cell separations per month. As they have to isolate up to 5 different subsets per patient, they request a bit more blood as compared to the previous centers we've discussed. They receive up to 24 mils of blood from adults, up to 8 mils from children, and of course they receive much less bone marrow. They FICOL their blood and start with both the PBMC fraction and the PMN fraction. Their accepted purity is 90% for all subsets, except for CD34, which is 80%. Again, they don't require a lot of DNA for their downstream assay, only about 2.5 nanograms, and they're using the ABI identifier STR kit for their chimerism testing. Their cell isolation protocol is a bit complicated, so bear with me. They start with whole blood, which they FICOL. They isolate the PBMC fraction and resuspend it in 1 mil. They load this onto RoboSep and isolate B cells using the CD19 positive selection kit. They then take the negative fraction, wash it, and split the sample. They load half on the machine for CD3 positive selection to isolate T cells, and the other half to isolate NK cells using the CD56 positive selection kit. They then take the gran layer above the red blood cells from the FICOLed blood and use it to isolate myeloid cells using the CD15 positive selection kit. So in this way, they can isolate B cells, T cells, NK cells, and myeloid cells in 2 RoboSep runs. As I mentioned, they occasionally need to isolate CD34 positive cells. When this is the case, they follow the same general protocol, but they do their CD34 isolation directly from the PBMCs. They like the RoboSep as they can assess purity by flow cytometry, and the purities are generally excellent. There's no cross-contamination between samples, and the machine requires minimal maintenance. There are no buffers that need to be made up daily, no tubing to clean, etc. The only maintenance is a water bottle that needs to be filled every 20 to 30 runs. In addition, the RoboSep saves tech time, it's easy to use, and it works well for low start percentage samples. They were able to validate the isolation of the 5 different subsets on 5 patients, and validation took them about a month. And when I asked if chimerism testing generated revenue for the lab, the answer was a resounding yes. So in summary, we've highlighted 3 different groups who brought chimerism testing in-house using 3 very different approaches. While each group isolates the same general cell types, T cells, B cells, and K cells, each has customized the RoboSep protocol to meet their particular needs. The Orlando group uses sequential separations to isolate 4 different cell types from a small volume of blood. The Children's Hospital of Philadelphia splits their samples as they only usually need T cells and NK cells. And as the Stanford group needs to isolate 4 to 5 different cell types from literally hundreds of samples, they've customized an approach that allows them to use the 3 machines to their best effect. Once the cells are isolated, the downstream DNA and chimerism tests are very straightforward. So I think what this demonstrates is that there's no one-size-fits-all protocol when it comes to chimerism testing. Cell isolation can easily be tailored to suit each individual lab's requirements. Some further pieces of advice for anyone wanting to start this in their own lab. First, to check the white blood cell or subpopulation counts before doing the cell isolations, as obviously, say if a sample doesn't have many T cells to start with, you can't expect to get many T cells out. You should always check the purity of your sample and be aware of antibody blocking. For example, the CD3 antibody we use in our CD3 positive selection kits is often blocking, and so T cells may need to be assessed using alternative markers. And importantly, you should check the yield of each sample, as this will determine the success of the downstream chimerism assay, which is of course the most important part of this whole exercise. So, is it easy to bring chimerism testing in-house? To paraphrase Kurt Lind from a conversation we had back in October, of all the HLA assays out there, this was the easiest to bring in-house. The cell isolation is straightforward, and the STR kits are very robust. And Kurt feels that every HLA lab has the necessary expertise already in-house to do this. So I'd like to stop there and open up the whatever computer line for questions for our panelists. And so I'll thank you very much for your attention, and I will turn it over to my colleague, Dr. Vicki Strong.
Thank you very much, Karina. That was a great presentation, as always. And thanks to everyone for hanging in there, because I know that we're running quite late today. So I'd like to now introduce our panelists again, Kurt Lind from Children's Hospital in Philadelphia, and Wendy Leong from the Stanford Blood Center. Hello, Kurt and Wendy.
Hello.
Hello. Great. So I'll just like to remind everyone, if you'd like to ask any questions, please use the panel to the lower right-hand side of the presentation. We have some that have come in already, and so I will start asking them. So somebody asked, can you get enough cells from pediatric samples using RoboSep? So I'll direct that question to Kurt to start.
The answer to that is yes, provided that the white cell count is sufficient. So there's not really any reason why it would be any more difficult with pediatric samples, other than the fact that they typically don't want to collect a lot of blood. So we try to give them the specimen requirements. We try to give them the bare, the minimum that they really need to collect. And before we do any isolation, we always check the white cell count, and as long as the white cell count isn't extremely low, we typically don't have any problems getting the cell population we're going after.
Great. And Wendy, can you get enough cells from pediatric samples?
Yes, we do. As a matter of fact, I concur with Kurt wholeheartedly. Since we do a preparation of high-pake PBN, peripheral blood lymphocyte density gradient, we have started to evaluate by cell counts how many PDN how many peripheral blood lymphocytes and total white cells do we actually have from that enriched fraction. And I'm very happy to say that we have more than 1 million cells in most cases, even in the pediatric patient, which we get typically not even a full 10 cc yellow top ACD tube. So the answer definitely is yes. We have not had in the history of our processing our pediatric patients for chimerism have very, very rarely been unable to report a chimerism result.
Oh, great.
It's been very rare.
Oh, good. And someone's just asked a question while you were speaking and saying, so what is considered to be a low white cell count for the system? And Wendy, will stay with you.
Well, typically we have not been in the habit of counting our mononuclear layers until fairly recently, within the last couple of years, well, last year I should say. And we have not had any difficulty in isolating. I can't tell you specifically what the count, what is the lower range. I don't know whether Kurt could answer that better than I.
I would say that what I consider to be low would be a 0.1 white cell count, so 100 cells per microliter or 100,000 per ml. So when we get down to that level, it's not uncommon for us just to not get the amount of cells that we need. So I wouldn't say it's extremely rare that we don't get enough cells in order to do the testing, but I would say that when, for example, we just had a sample that was still a low white cell count, so a 0.8 white cell count was still sufficient. We got from even 1 ml of that blood at that white cell count, we were able to get enough DNA to do the chimerism testing.
Great. Okay, thanks, guys. So someone also asks, do you use the myeloid kit that separates CD33 and 66B, or just CD33? So either of you that's using the myeloid kit, we'll start with Kurt.
So we don't do a lot of myeloids currently, but when we do, we're using the combination kit. So it's positively selecting for CD33 as well as 66, and so that group is including monocytes and granulocytes. Now, just within the last week, we have a case where the physician's interested in looking just at the monocytes, and so we didn't have that in house. So last week we found that there was a CD14 positive selection kit. We ordered it. We got it in at the last, at the end of last week. We're in the process of validating it, and we'll be able to, using a different kit, we'll be able to look just at monocytes. And so it's nice because, all of these kits are, they're really interchangeable. And so if you get these oddball requests from time to time to look at a particular cell type, it's really pretty simple to bring it in, validate it pretty quickly, and go forward with it.
Great. And Wendy, your myeloid choice of separation?
Is right now the CD15. It's been the main myeloid choice for the myeloid series. We had done some experiments, or had done a research project using the CD33 selection kit and separately the CD66B kit. We did not use the, we've not had any experience with the combined kit yet. And we have had experience also in the past with the monocyte kit, and Kurt, it does work very well. We use it for right now in our laboratory, the CD14 for another purpose, but we don't use it for the chimerism purpose right now. So.
Okay, fantastic. So here's an interesting question. Has the interest in subset analysis for chimerism been driven by your clinicians, or is there an educational effort needed on the part of your labs? So essentially, has a strong clinical relevance for subsets been established, or is this in process? So I'll direct that one to Kurt.
Yeah, I would say it's been driven by our clinicians at this point. We let them know when we first had that capabilities, and they were very much interested, particularly with T cells. Depending on the protocols they're using and the different regimens they're using on these patients. So our, the number of, or the percentage of samples that we're doing lineage-specific analysis on has increased since we've begun. But I would say it's driven by the clinicians.
And Wendy, do you have any comments on that question?
I agree with Kurt that it is mainly driven by our clinicians. They use this, the chimerism results that we generate, not as a total diagnostic tool, but as a adjunct to their clinical findings. And as a result, they are able to tailor the type of therapy to either rescue a graft. Typically, if they notice from our results and with their own clinical results that a patient is rejecting a graft, they will be able to jump in and tailor a particular appropriate therapy, be it donor lymphocyte infusion, boost, or else if the situation warrants a second transplant. But they find that the information we provide is very useful in the treatment or the clinical outcome of the patient's post-transplant experience.
Mm-hmm. Okay, thanks for that. And someone has asked, what issues, if any, do you encounter with samples that are older than 1 day? So I'll direct that question to Wendy again.
Yes, we have in our protocol, our test has been validated for blood being no more than 48 hours old, and marrow no more than 1 day old must be processed. What happens is, in the process of our validation, why we made that caveat, or we make that statement on our reports, if a specimen is like drawn on a Friday but doesn't arrive in our laboratory until Monday or something like that, is that we found that it does have an impact on the results of the chimerism testing, simply because the viability of the cells are not as good if they are, say, 72 hours old or greater than 48 hours old. Mm-hmm. And it can skew the results because the dead cells still and have the markers. They may be just floating free in the system, and you don't know whether those the markers that you're looking for are truly from the cell population you're looking for. Mm-hmm. Because other cells, like some of the dead cells, may have the marker that, say, CD3 on the cell surface, but they may, like NK cells, also have CD3 on the cell surface. So how do you know if the cells are dead whether the results are reflecting what the live cells would have reflected? Am I making myself clear, or is that?
Yeah, and someone's just asked, aren't dead cells eliminated with positive selection? But my guess from your answer is that no, because they still express the markers that you're using to select the cells. Is that right?
Yes, that's correct.
Okay. And Kurt, what issues do you have with any older samples? Is it viability as well?
Yeah, I would be hesitant to accept anything that's greater than 48 hours old. and we don't really have that problem here, so we're not trying to test the limits of what's acceptable. But yeah, I think that it's, just to kind of go back to the question as to whether or not you would or would not select these cells if they were dead, as long as they're intact and still are expressing that CD marker, then you would still select them. And we know in an HLA lab that not all cells die at the same rate. I mean, these cells are much more sensitive and tend to die before other cells. And if they start to fall apart, then you could have kind of a different balance of your differential, your white cell differential. So we haven't done a lot of work in this area to kind of test the limits of how far out we could go, but just for us, I would say anything over 48 hours we would not accept, and we'd ask for a new specimen.
Okay. Excellent. So I'm going to keep going with the questions. I appreciate if people need to leave because, of course, you've got a busy schedule, except the panelists. I hope you can stay. But just want to let everyone know you will be able to receive the full ABHI credits if you're not able to attend the right up to the very end. And so we have a question, what conditions do the experts recommend for sample shipping transport, either bone marrow or blood? Is there any usefulness in receiving frozen PBMCs, or is separation still possible with frozen PBMCs? So I'll ask Kurt if he has any experience with that.
I would not. We don't receive anything that's from outside, and I would not accept anything that had been frozen.
Mm-hmm, mm-hmm.
I'm curious as to what Wendy thinks about that.
We too have not received frozen cells to process, and even in-house we do not do anything on the RoboSeps with frozen blood. We have had DNA, of course, shipped to us from other laboratories to perform chimerism on, and that's been no difficulty whatsoever. But as far as unfractionated or differentiated cells for us to fractionate, we have not accepted any samples like that. Typically, we have received blood from far away within the United States, like cross-country, but those are shipped to us in the typical quote-unquote kit, which includes the ACD tubes, the solution A ACD, I might add. There's solution A and solution B, as all the HLAers know, and solution A, acid citrate dextrose with a 10 cc size tube is the specimen of choice, and we have them send back the sample overnight express mail, and we will process it immediately upon receipt, and we've had very good success with that.
Okay. So fresh is obviously preferred by our panelists. We do indeed have kits that can be optimized for frozen PBMCs. So if you're in a different area, your samples are coming from far away, then we certainly have solutions for that, and you would just need to make sure you talk to us at Stem Cell to optimize the protocol for your samples. Okay, so someone has asked, what are the ABHI guidelines for purity of subpopulations at this point based on clinical relevance? Is there any experience or success with repurification in the instance where purity is low, so below 50 to 80%? So there have been a few questions about purity. Where do we see any low purity? What's done about it? But perhaps, Wendy, you might answer the question about the ABHI guidelines for purity, if there are any that you're aware
of. We have, as far as specific ABHI guidelines on purity for STRs, I'm not really sure whether they specify. I know ABHI guidelines dictates that we must have some sort of purity designation, and what our laboratory says is acceptable for purity is, as Karina had indicated, is greater than 90% for the majority of the subsets that we have or the in our in-house QC, that when all lots of the stem cell technologies product or any product of our immunomagnetic beads are tested, greater than 90% for all subsets, with the exception of CD34, which is greater than 80%. But I will say that in practice, the majority of our purity upon QC is greater than 95%. And for CD34, it's greater than 90% rather than greater than 80%. We have received some lots that have been a little bit lower than that, for CD34, lower than say, the 90% purity, but definitely above 80%. So it is, as far as individual patient purity, we do not currently evaluate individual patient purity. We go by the guidelines of what our laboratory has established, and for acceptable purity upon QC of each lot of beads that we receive. So we don't accept anything less than that. So nothing less than the 90%, or in the case of CD34, 80%. That's not acceptable.
Mm-hmm. And
there, yeah.
I'm just curious. I'm gonna keep asking, because I'm sure this colleague might want to know. Is there any experience when you do experience low purity? Is there anything you can do? Any repurification?
Of the beads, or are we talking about individual patient, or?
I think that I think the person might want to know about if there is an experience with low purity. It doesn't sound like you do have that, but if it did happen, is there anything you can do to rescue the sample?
We don't have anything to rescue the beads. We just say that that is unacceptable, and get a new lot of beads to isolate.
Okay, and Kurt, yeah.
Or the test sample. Perhaps if we're talking about QC and you find that the specs come in less than 90%, or that are really low, meaning 50, 60%, then I would question what cells are you using for the QC. Are you using dead cells, or is the viability of the sample at issue? Or do you have a patient that, are you using a sample for your QC that is from a donor or donors, a pool of donors, that something's abnormal in there?
Mm-hmm. And Kurt, do you have any comments on purity guidelines from ABHI, or perhaps your experiences with the purity?
Yeah, as far as I know, looking at the most recent ABHI standards, they don't. ABHI doesn't say that you have to hit a certain purity. In fact, it's not even at this point. I think this may change, or I think there's some discussion about requiring that purity assessments are done on each sample, because at least initially when we started doing it, ABHI didn't even require that you did a purity assessment for each sample. We do that ourselves, but my understanding is that not everyone does that. Not everyone has access, maybe to a flow cytometer to do that. But the ABHI standard says that you need to document the purity obtained if processing involves an isolation of cell subsets, and that if purity is not assessed, then you need to indicate that on your report. As far as what we do if we don't hit, actually, our criteria is 90%. We've since adjusted it after having had more experience now using the RoboSep. Our internal QC, meaning what we consider to be a valid isolation, is greater than 90% of the intended target. If it's less than that, we we typically would go back to the initial specimen if we still have some. We've made some changes to the protocols where we are using just maybe 1 ml of whole blood and not using the full amount. So we can go back and start with a larger starting volume and try to increase our chances of pulling out the cells that we're going after. And typically when we don't hit purity, it's because the cell count is really low. Otherwise, we're over 95% almost all the time. When the purity drops, it's typically because the cells just simply aren't there in sufficient numbers. So we can go back and try to re-enrich. But then, in some rare instances, we simply have to report it out and say there simply wasn't enough cells there for us to enrich and get 90% purity.
Great. Okay, and so in doing the purity assessment, so running by flow, someone is asking, do you see any morphological changes on the flow analysis for any of the cell types that you separate? And this person is particularly interested in the CD14 positive population. So Kurt, I don't know if you have an answer for that.
Well, I don't have any experience with the CD14 yet, but as far as differences, the way the cells appear on flow cytometry, they look nearly identical. I mean, the beads themselves are very small, and so when you look at light scatter, properties, forward scatter and side scatter, the cells typically show up where you would expect them to show up had they not been captured with beads. Mm-hmm. And so what I'm getting at here is that many of us are, comfortable or we're familiar with where lymphocytes typically show up, or monocytes typically show up, or granulocytes typically show up when we look at forward scatter versus side scatter. And that remains to be the case with cells that have been isolated with these beads because the beads are so small. Which is significantly different than beads that you get from another vendor. Many of us have used beads from other vendors for cytotoxicity assays, and those beads are considerably larger. And when you try to run those cells that have been isolated with those other beads through a flow cytometer, it's pretty messy. So the data looks pretty clean on the flow. We typically do a two-color analysis for purity. We stain with CD45 so that we can just gate on those cells that we know are white blood cells. And then our second color is an antibody against the targeted CD marker.
Okay, and Wendy, do you have any experience with looking at the cells by flow, any morphological changes after separation?
As far as morphological changes, no, I don't note anything. I concur with Kurt.
Great. Okay, a couple of quick questions. Someone asked if we QC on normal samples or patient samples in-house. We at Stem Cell do our QC on normal samples. So, and Wendy, but Wendy may have experience doing some QC of our kits on patient samples.
For the source of CD34, we've had to use marrow, and simply because we don't get enough cells for CD34 with typical pool, even if we pooled from peripheral blood of normal donors. So that would be the only instance that we use patients for. Otherwise, most of our QCs are done on whole blood from normal individuals. But there's no problem. Even with patients, we make sure that the patients are in they're not in remission or relapse or something like that for those marrow samples that we use.
Okay. And Kurt, do you have any comments on your performance on normal samples or patient samples?
We do our QC typically with normal samples, but I'd say that from time to time we probably do some QC on patient samples as well, and again, as long as the white cell count is decent, you know, we tend to get the same recovery and the same purity, whether it's a normal or a, you know, or a patient sample.
Great. Okay, so unfortunately we're running out of time. We're at about 60 minutes now, so I'm sorry if I didn't get a chance to get to all of the questions, but we'll certainly be able to answer them personally, and we'll consult with our panelists if they don't mind contributing to the answers so that they can, people can gain from your wisdom. So I just want to say thank you everyone for attending this webinar. Thanks for bearing with us through our technical difficulties. A very big thanks to Kurt and to Wendy for participating in this webinar and answering your questions, and of course to Karina for a fantastic presentation. Thank you very much, and have a great day.